right? To secrete what?
Bile.
To secrete bile. Which will be stored where?
The gallbladder.
Which is stored in gall. Gall what? Gallbladder. Gall. It's gall. Not gall for gall. It's G-A-L-L. Gall, blood, right? Of which at another point what happens? What happens again there? Pancreas does what? Yes?
Secrete the digestive enzyme.
We have again the pancreas to secrete those pancreatic what? Juices. That's clear. Pancreas to do what? To secrete pancreatic what? Juices. What about now the cholecycline? It stimulates that liver
The gallbladder.
to release.
The bile.
Oh, not liver, sorry. It stimulates that gall what? Blood to secrete what? To release
Bile.
the bile juice. Is that clear? So we are saying this one is the one that stimulates the liver.
Yes.
Right? Those are the peptic cells within the liver to secrete what? Bile. Now here we are speaking about it was stored in the gallbladder. This cholecycline stimulates that what? Gallbladder to release the bile juice. Is that clear? We are saying it stimulates what?
The gallbladder.
Gallbladder to do what?
To release bile.
To release bile juice. To which point? That duodenum area, right? We have another one?
Hiyo hapa Chris. Kwa hizi notes ni kwa hii ingine, sio kwa hiyo ya secretive.
I'm speaking about the two roles here. Do you understand? This is what I'm speaking. I've said this one secretin stimulates liver cells to secrete what? Which is part of the gallbladder. Cholecycline, I've said is what? Pancreas to secrete pancreatic juice. Then number 2 stimulates this gallbladder to do what? To release bile juice. Is that correct?
Yes.
Yeah, that is it. Then after now that point, what happens now? We go about our digestion at this point. What will be the role of bile? We have roles of what?
Bile.
Bile juice will have some 3 roles. It will have 3 what? Roles. Number 1, neutralization of what?
Acid.
So you know our food, not our food, but our child is from where? Stomach. Within the stomach it was having what? Hydrochloric acid. So it will neutralize the acid. Is that clear? Neutralization of what? Of the acid. Is that clear? From the stomach. That is number 1. Number 2,
Provide suitable alkalines.
provide the alkaline conditions. And you will understand the 2 components of the bile salts. It will be having some salts. Sodium glycocholate and sodium taurocholate. Right? So those are the ones that brings about what? Neutralization. We are saying it will do what? Neutralize what? Neutralize the acidic chain. Acidic chain from stomach. And the last one?
Provide suitable alkaline medium.
Providing suitable alkaline.
Umerudia mara mbili.
Oh, this one was the acidic medium. This one was wrong. We are saying providing alkaline medium. Provide what?
Alkaline medium.
Provide alkaline medium. Medium for the pancreatic what? Pancreatic enzyme. So when you speak about this providing acidic alkaline medium, we are speaking about pancreatic enzyme. We have 3 pancreatic enzymes. Which are those ones? Pancreatic lipase, pancreatic amylase, and trypsin. We are saying pancreatic lipase, that is, it will deal with lipids, right? Pancreatic amylase will break the remaining starch. And we have trypsin will break the remaining what? Proteins. I get the point. So what will happen? This one is the one that provides that alkaline condition. Which one is the last role of the pine tree? Emulsification. We have what?
Emulsification.
What is emulsification?
Break down fats into
We break down the large fats into small fat droplets. That is what? Emulsification. And it will be done by the pancreatic what? Lipase. Are you get the point?
Yes.
Then we are speaking about the gallbladder will be released. So we have those pancreatic juices. How will our digestion happen now at this point? Let's speak now. We are not speaking about the digestion here, right? The digestion will be done. How will the digestion be done? We speak now about the proteins. So we are speaking about the pancreatic juices.
Yes.
Are you getting this point? We said we have 3 pancreatic juices, right? And I've said pancreatic juices are which ones? Pancreatic what?
Lipase,
Pancreatic lipase.
amylase,
Number 2.
pancreatic amylase,
Pancreatic what?
amylase,
Amylase.
tripes,
And what?
tripes.
Trypsin. Is that clear?
Yes.
Now let's go the digestion one by one. The food, that is the chain. It will be having those results of food, right? So we are saying, we are speaking about remaining. The only thing that we have not digested to the stomach is only one, which is that food component. That we have not digested from all to duodenum. I think we have digested the all except one. We have 3 food components. Which are those 3 food components? Carbohydrates,
Proteins,
proteins, and what?
vitamins.
And vitamins. Not vitamins. Vitamins are not digested. Vitamins are absorbed directly into the bloodstream. Are you get the point? Fats. Fats is another one. So I'm saying what? Which one have we not digested here? Fats.
Fats.
We have digested our protein. So you speak about like for starch. So you speak about remaining what? Why are you speaking about remaining? We have digested something where? Mouth. So if remaining starch is digested what? Broken down by what? Pancreatic what? Pancreatic amylase. To what? What are we breaking down? We broke starch to what in the mouth?
Maltose.
To maltose. Is that what clear? We are breaking it down to what? Maltose. Then you go to the second one, which is pancreatic what? Remaining what again? Protein. We break protein to what? What are we breaking proteins to in the stomach?
Peptides,
Broken by what first?
tripes,
Pancreatic. Trypsin. Is that clear?
yes,
To what?
pep,
To peptides. Peptides. Is that clear?
yes,
We are breaking into peptides, this will break to maltose. Then now we have the first digestion of lipids. We said lipids is not digested yet. This one is our first digestion process, right? So we are breaking down, so it's not remaining, right?
yes,
Breakdown of what?
lipids,
Down of lipids.
fatty acids,
To what?
fatty acids,
To fatty acids
and glycerol,
and glycerol. By what?
pancreatic lipase,
By pancreatic
lipase,
lipase. By what? Pancreatic lipase. And that is the end of our digestion in the duodenum. And we say yes. Float. Let me go again. Now float together watu wewe. Haya, don't float. And don't sing. So what you should know what? This is what we are speaking here. Let's start from the duodenum. We are saying from the duodenum what happens? The food enters the duodenum. It's not food, sorry. China. Enters the duodenum through what? Pyloric sphincter. When this now, in small amounts at a time. And that is why the role of a pyloric sphincter is so that we can still retain. So you know there is digestion in the stomach. We still give more time for digestion in the stomach. And allow that some to the duodenum. So that the digestion be what? Progressive, right? Then at this point, when the chime is in the duodenum, is that clear? It stimulates what? Secretion of these pancreatic juice and pine juice. And you'll get a question asking you, Why is duodenum? Is it duodenum? Yeah, duodenum. Described as an endocrine and an exocrine. It's endocrine because we will do what? Because of the secretion of the pancreatic juices. It's not duodenum, sorry, pancreas. Pancreas is what? It's described as an exocrine and what? An endocrine. It's endocrine because of the secretion of the pancreatic juices. Is that clear? It's exocrine because it has a digestive role. Digestive juices which will do what? Bring about the digestion. Are you get the point? Endocrine and what? Exocrine. So let's go back to this one. So you understand how this one? We secrete two things. Pancreatic juice and what? Bile juice. The duodenal wall now, again, stimulates secretion of secretin. It's about the child that stimulated the pancreas. This one is secreted by what? Pancreas. Now, so what's the child? Duodenum, it stimulates the pancreas. Oh, I've said just secretion. In a stimulate what? You have pancreas here, is that clear? It stimulates the pancreas. That's not happening. To secrete the pancreas juice and what? Bile juice. And that's why in the pancreas will be what? Will be described as what? Exocrine.
Endocrine.
Endocrine is because of the pancreatic juice. Here again. Exocrine because of the bile juice. Bile juice will go and do around
Digestion.
right? Then next now, the duodenum stimulates. It's stimulated to secrete secretin. That's the duodenal wall of the duodenum. The other one was in the stomach. It was gastric gland. Being stimulated to secrete what? Gastric juice. Here is our duodenal what? Wall. It's stimulated to secrete what? Secretin and the color what? Cytokinin. Then this one secretin, we are saying it stimulates the liver. And that is why we are circulating with many organs there, right? From pancreas, we have our duodenum. We have now what? We have the liver. Now this secretin stimulates that liver. So digestion process will involve all your organs, right? It now stimulates your liver cells, which are known as peptic cells, to secrete what? Bile, which is stored in where? Gallbladder. Peptic cells will be stimulated, the light of peptic cells. The peptic cells will stimulate what? Secretion of that hydrochloric acid. You'll be asking, how will the peptic cells what? What secretes hydrochloric acid? It's the peptic cells in where? The stomach. Then stimulate the liver cells to secrete what? Bile, which is stored in where? Gallbladder. Then the second thing here is what? Cholecycline. This one stimulates that pancreas again to secrete those pancreatic what? Juices. Then it stimulates gallbladder to release what? Bile juice. I'm saying this is just a repetition of this one. Don't you see that? It's just a repetition of what we are speaking. So it's about the duodenal wall which will be serving those functions. To do what? Secrete two things. Cholecycline and secretin. So that secretin stimulates that liver to secrete what? The bile, which will be stored in the gallbladder. Right? These are the one is the cholecycline to stimulate that liver. Not liver, the gallbladder, which has stored the what? Bile juice. To secrete what? Those or to release those bile juice. Then again it stimulates that pancreas to do what? Secrete the pancreatic juices. And our pancreatic juices is what will take us here. But before that we'll start with our what? You have to start with bile before this one, because this one is the one that will function for, will be released for this one to function. Is that clear? So you now go to the bile juice. You'll be asking this one as an independent question. What is the role of bile juice? Number one, neutralization of the acid. And I've said why does it do neutralization? It will be having some two salts. Two salts of the bile juice are which one? Sodium glycocholate and sodium taurocholate. I get the point? Glycocholate and what? Taurocholate. Then that is number one, neutralization. Number two, providing an alkaline what? Medium. Then number three, and then by for this pancreatic what? Enzyme. Then number three, emulsification. And we are saying emulsification is what? Break down what? Large fats to fatty acids and is it fats and glycerol? No. Large fats to fat droplets. Large fats what? Fat droplets. Then now we are speaking about the pancreatic juices contain the three components. Pancreatic lipase. Are we going with it? Pancreatic amylase. And what? Trypsin. Then this now we are speaking about is about breakdown of remaining only except what? Lipids. We said the lipids will be the first breakdown. This pancreatic, the starch, will be broken down by pancreatic amylase. So we have two amylase enzymes. Which are those? Those two amylase enzymes. Salivary amylase enzyme and the pancreatic amylase. Salivary amylase, no. Pancreatic amylase, duodenum. Is that clear? Break it down, remaining what? Starch to maltose. That is one. Number two, remaining proteins will be broken down by pancreatic what? Pancreatic sorry, we don't have something to do with pancreatic trypsin. It's broken down by trypsin. Is that clear? To peptides. We don't have pancreatic trypsin, it's just trypsin only. Is that clear? It's broken down by what? Trypsin to what? Peptide. And that is why when you write your exams, you have to do what? You have to reread. So that you check mistakes and you understand that I've done wrong. Is that clear? So I've just been writing fast and that's why I've made a mistake. So it's trypsin to what? Peptide. That breakdown of the lipase now will be done by what? Pancreatic lipase. Where it will break those fatty acids, the lipids, to the fatty acids and glycerol. And now that's where our emulsification becomes different with this one. Emulsification, we are breaking down what? Large fats to fatty droplets. That is it. Is that clear? Then lipase will break down the lipids to fatty acids and glycerols. I get the point. And that is the end of the digestion in where? Duodenum. Then now, the other one I'm not right, I'm just using it, I'm making it to finish. That is a which point? The ileum. Ileum is still part of the small intestine. Small intestine is divided to what? Two parts. Duodenum, ileum. Ileum, that is where we have complete digestion. Is that clear? Then duodenum, there was a digestion. And now here, let's complete our digestion. What happens in the duodenum? At the duodenum now, the food, the child, will leave to the ileum. It will form something already called the child. Is that clear? Then now, from this point, the one when it enters where? When it enters the ileum, what happens in the ileum? There is complete conversion. It's no longer digestion. Is that clear? It's about what? Conversion. And what brings about this conversion? There are the enzymes now. We have the interstitial what? Secretion of the interstitial juices. Is that clear? We have the interstitial what? Juices. In scientific, we call it suckers and triggers. Suckers and triggers. And when you write suckers and triggers, it should be follow the binomial nomenclature. Is that clear? So just say what? Those enzymes are secreted from which one? Not suckers and triggers. Interstitial cells. Never. Like the now pancreatic lipase will be broken down still by the pancreatic lipase. Maltose, you still remember about factor of enzymes that I said what? P, not pH specific, but what? Enzymes are what? Substrate specific. Maltose will be broken down by maltase. That's now the specificity content coming up there, right? Maltose, which is here, is broken down by what? Maltase to what? Glucose. That is one. Let's build first with carbohydrates. Carbohydrates can have a wide range. Even maybe your food adds sucrose, right? So that sucrose will be broken down by what? Raised to fructose and what? Glucose, right? Then you can have lactose. Your lactose will be broken down by what? Lactose is in milk. Maybe our child took milk. So the milk sugars are what? Lactose. They are broken down by lactase to galactose and glucose. I get the point? We are done with the what? For the proteins. For the not for the proteins, but what? For carbohydrates. For proteins, we said we have what? Peptides. What will break down the peptides? Peptidase enzyme. Peptidase will break peptides to amino acids. I get the point? Peptidase breaks what? Another name? Peptides to amino acids. Then now the last one is what? Lipids. It will still be lipase. For lipids don't change. The others have changed. For lipids don't change. It's still what? Lipase. It will break what? The remaining. You see the first point was what? We just started our digestion of lipids at the duodenum. So it's still the remaining. We just convert. What happens here? The lipids will be broken down by the lipase to what? Still those fatty acids and glycerol does not change. That way. We are done with what? Conversions and digestion in the ileum. Is that clear? You go something called assimilation. What happens in assimilation? Assimilation is taking those digested food into the body cells, right? We are assimilating which point? Still in the ileum. Is that clear? So what happens in the assimilation process? So we are saying you have glucose. Glucose is assimilated to the body, right? It's assimilated to the body. What? Then we have what again? To the body cells. Assimilation is what? Taking those digested to the body cells. So we assimilate glucose, we assimilate amino acids, but lipids cannot assimilate to the blood. We don't take lipids to the blood. We take them to the lacteum. So you know the lacteum? The filler structure. What happens in the filler structure? We have something here called lacteum. That is where we take what? We assimilate our lipids to this point, to the lacteum, where it will be taken to the lymphatic system. These other ones are taken to which point? In our blood, we take glucose and the rest to which point? To the rest parts of the body, so that we can take them again back to the liver, so that we can regulate the excess blood sugar levels, right? From that point, the remaining digested and the undigested food will do what? Be taken to which point? To the rectum. No, first is the large intestines. In the large intestines, there is absorption of water. We don't have digestion in the stomach. We just have absorption of water and even synthesis of some vitamins like vitamin K. Is that clear? Then from that point, they are empty to which point? To the duodenum, where they will be temporarily stored and they will be eliminated out of the body through which point? Through the anus. And that is a complete. That is now from ingestion to ingestion. Thank you for having me in this lesson today.